Good morning, everyone. First of all, many thanks to the organizer to giving me the opportunity to present this work that has been performed in Servier Research Institute in France under the direction of Dr. Marie-Pierre Bourguignon, that is unfortunately not able to come and present this work by herself. Aging is associated with vascular dysfunction that leads to atherosclerotic development. This is a long-lasting process that is characterized by an alteration of the vascular reactivity. This vascular reactivity is leads by the endothelium, that is the inner layer of the vessel wall, that integrates the stimuli from the blood flow and transduces them to smooth the underlying smooth muscle cells that will relax or contract in order to adapt the vessel diameter to the needs of the surrounding tissue. This vascular reactivity is very important for the vessel homeostasis, and the age-associated loss of vascular reactivity is a consequence of the loss of endothelial function. This endothelial function, or the endothelial dysfunction, occur with age and is when it's associated with risk factors like diabetes, hypertension, hypercholesterolemia, and they are all associated with an increase in oxidative stress in the vessel wall. And it leads to the appearance of atherosclerotic plaques that are responsible for a big part of the cardiovascular diseases. It induces ischemia in brain, myocardium, kidney, or in periphery. When there is a plaque rupture, there is stroke in the brain, myocardial infarction, and myocardial ischemia and myocardial infarction are responsible for the appearance of heart failure. Among the different vasoactive molecules that are synthesized by endothelium, the nitric oxide support a big part of the endothelial function. They are all listed here, and most of them are protective against the appearance of atherosclerosis. In this study, we will be focused on vasodilation that is representative of the whole endothelium function. This nitric oxide is synthesized by the endothelial nitric oxide synthase. This is an enzyme that needs tetrahydrobiopterin, BH4, as a cofactor to work properly. In normal condition, when BH4 levels are normal, the enzyme is using oxygens and L-arginine to produce nitric oxide. But when the BH4 level is reduced, the dimer of nitric oxide is less stabilized, and the enzyme uses oxygen to produce superoxide anion. This situation where the nitric oxide production is reduced and oxidative stress is increased has been described in many cardiovascular pathology and is associated with most of the risk factors that I cited in the previous slide. The BH4 is synthesized by a series of enzymatic activity, and the rate-limiting enzyme in the GTP cyclohydrolase 1, GTPCH. And when BH4 has been produced, it can be rapidly oxidized into BH2. The BH2 is also able to bind nitric oxide synthase, but when nitric oxide synthase bind BH2, it is in the uncoupled state, that is to say, it produces superoxide anion instead of nitric oxide. Taking all this information into account, we wondered what could be the role in the reduction of BH4 that occur because of oxidative stress associated to risk factor of atherosclerosis in vascular dysfunction that occur with age. And we wanted to see what is its role in different kind of vessels. For doing so, we use a model of BH4 deficiency, that are the HPH1 mice. It is a model with a 50% reduction of GTPCH activity, and of course, a reduction in BH4 levels. At the beginning, this model was a model of hyperphenylalanine because BH4 is also a cofactor for phenylalanine oxidase. But during his lifetime, these animals have a lot of cardiovascular dysfunction that are listed here. We used young animals that were 12 weeks old versus middle-aged animals that were from 35 to 45 weeks old. And we measured the vasoreactivity in 4 types of vessel: aorta, mesenteric arteries, coronary arteries, and cutaneous microcirculation. For all these vessels, we choose what we considered the best stimulation to induce a nitric oxide dependent relaxation, that is to say acetylcholine for aorta, the flow mediated dilation for mesenteric artery, that is to say the adaptation of the vessel to an increase in blood flow, the hyperemia response for coronary arteries, that is to say the vasodilation of the coronary artery in response to a transient ischemia. And for cutaneous microcirculation, we tested, like for the mesenteric arteries, the flow-mediated vasodilation. And we also choose what we consider the best technique to measure the vasoreactivity. That is to say, for aorta and mesenteric arteries, isolated organ chambers. For coronary arteries, it was a isolated Hart-Langendorf technique. And for the cutaneous microcirculation, we used a laser Doppler flow meter. We first verified that the level of biopterin, and especially BH4, was efficiently reduced in the mouse model of BH4 deficiency. We were quite surprised at the beginning to see that the BH4 level in the aorta was increased in middle-aged animal compared to control and young animal. But this is certainly a consequence of what have been already described, that is to say that GTPCH, the rate-limiting enzyme in BH4 production, its activity is increased by oxidative stress. And that certainly is the case here, because when you look at BH4, the oxidized form of BH2, the oxidized form of BH4, we also see an increase, and this increase is bigger than the one for BH4, suggesting that there is a global increase in biopterin production, and a part of this biopterin production, BH4, is rapidly oxidized into BH2. In the animals that were BH4 deficient, we have a reduction, a huge reduction in BH4 level in the aorta, either in the young animal or in the middle-aged animal. This is associated by a reduction in BH2 only in the middle-aged animal. And this reduction in BH2 is smaller than the reduction in BH4, once again showing that a big part of the BH4 that is produced is rapidly modified into BH2. We measured the cyclic GMP in the aorta, because cyclic GMP is the second messenger for nitric oxide bioavailability. And we saw that the modulation in BH4 and BH2 production in normal animal has no impact in the basal nitric oxide production. And the reduction in BH4 has no effect in young animals, whereas it induces a clear reduction in basal nitric oxide production in the middle-aged animal. We thus wondered to know if there is a consequence in nitric oxide dependent vasodilation, that is to say in the production of nitric oxide when the nitric oxide synthase is stimulated. And we saw that in young animals, there is a clear vasodilation dependent of acetylcholine concentration. This vasodilation is totally abolished by an inhibitor of nitric oxide synthase, showing that it is a NO-dependent vasodilation. And the BH4 deficiency in this young animal has no effect on the vasodilation. In the middle-aged animal, in control animal, we have the same vasodilation as in the young one. And the BH4 deficiency induces no dysfunction. We then looked at mesenteric arteries. We perform an experiment to see the flow-mediated vasodilation. And in young animals, as the flow increases, the vasodilation increases. In the middle-aged animal, normal animal, middle age, we saw a slight reduction of the flow-mediated vasodilation, and this slight reduction clearly amplifies when in the animals that have also BH4 deficiency, whereas the effect of BH4 deficiency in young animals is here, but is not significant and is quite limited. In middle-aged animal, we also look at the coronary artery vasodilation by using a Langendorf isolated heart experiment. We stopped the flow through the coronary tree, and when we restored the flow, we studied what is called the hyperemia response, that is to say, an important and rapid vasodilation of the coronary artery that goes up to its maximum, and then a second phase when the flow, so the vasodilation, decreased to a normal value. And what we saw in this middle-aged animal is that in the BH4 deficient mice, there is a reduction of this hyperemia response, especially a reduction in the peak, that is the maximum vasodilation, also called the coronary flow reserve. And there's also a decrease in the global area under curve. So here again, in middle-aged animals, the reduction in BH4 level induce vascular dysfunction. And the last type of vessel we studied were the cutaneous microcirculation. In this case, we used a laser Doppler flowmetry to measure the flow in response to injection of acetylcholine. You can see the clear vasodilation induced by a repetitive injection of acetylcholine. And in the BH4 deficient animals, we saw that this vasodilation is clearly reduced up to 70% of reduction. And here again, in the middle-aged animals, the reduction in BH4 level induces a clear vascular dysfunction. The conclusion of this study is that in the aorta, that is the only conductance vessel that was tested, the BH4 deficiency is able to limit the basal nitric oxide production in the middle-aged animal, but it has no effect when the nitric oxide is stimulated to induce a vasodilation. But in the three other type of vessels that are all resistance arteries, the BH4 deficiency in middle-aged animal induce a reduction of the vascular reactivity, and it seems that the smaller the vessel are, the bigger the vascular dysfunction is. In conclusion, the BH4 deficiency is able to induce a vascular dysfunction in aged animal, in peripheral and deep microcirculation, as it has been clearly described that in cardiovascular diseases, and especially associated with risk factors like diabetes or hypercholesterolemia, there is a huge oxidative stress in the vasculature. We know that the BH4 levels are reduced. So we could assume that with age and this risk factor, this reduction is BH4 certainly plays a role in the vascular dysfunction that leads to atherosclerosis, and it opens the door to a potential therapeutic solution by restoring BH4 level. And this is not quite something totally new, because in the 2 last decades, there were a lot of trials, clinical trials, to supplement with BH4. Some of them injected BH4 directly in the coronary circulation. There were also trials for supplementation by oral route. And even if some of them gave promising results, to date, it is not a valuable therapeutic solution, mainly because in these pathologies, the injected BH4 is rapidly oxidizing to dehydrobiopterin, BH2, and is longer able to give its protective activity. So there is still a lot of work to do to find a therapeutic solution. And the solution will be when someone will find a way to restore the BH4 level without increasing the BH2 level. Thank you for your attention.